Well, good morning and hello, everyone. Thanks for joining our webinar today on neurobiology research. I'm Sally Head-Delquist, and I'm president of Immunochemistry Technologies. At ICT, we're on a mission to help cure disease, and we do that by providing better assays to study cell viability. These assays help researchers understand the pathway of neuronal disease, monitor disease progression, determine efficacy of treatment, and that ultimately will lead to better treatment of neuronal diseases, and that's why we're all here, really, to help cure disease and save lives. I have a BS in genetics and cell biology and an MBA. I started out working in the lab, helping to develop these assays, and now I'm president. Assisting me today are Allison Westcott, our customer service manager, and Nick Wenninger, our scientific associate, who put together much of this data. So a big thanks to Allison and Nick for helping. And to those of you out there watching, we really appreciate you taking the time to make this webinar a part of your day. We've muted everybody's phones, but we really encourage you to ask questions. So just type them into the chat box or email them to help@immunochemistry.com, and we'll respond directly to you after the webinar. And also, we're going to be recording this, and we'll post it on our website and YouTube and email a link to you, so you'll be able to watch it again and share it with your colleagues. I'm really excited today to share some of the work that's been done on neurons. ICT's products have been used by researchers all over the world in hundreds of publications, and there's a lot of interesting ways they've been used to look at apoptosis, oxidative stress, mitochondrial health, and lysosome functionality in cultured neurons, in tissue sections, brains, live animals, and related cells in vitro, in vivo, and ex vivo. So in this webinar, we'll highlight these experiments and explore the techniques and models of disease the researchers have used to evaluate these conditions. And I really hope this webinar will give you some inspiration when setting up your next experiment. To get a little background, here's an image of two healthy neurons, an upper motor neuron and a lower one that connects to the muscle. Today we're talking about neurons, and the neuron's main function is communication, telling a muscle to move. But traumatic injuries, lack of oxygen, and many different diseases cause problems transmitting this signal and keeping the neuron functional. And there are many cells that protect and work with the neurons, like microglial cells, which are immune cells, but sometimes can cause damage if overexpressed. And it's a complex system, but fortunately we have a lot of smart people researching these diseases, which makes me optimistic we'll find better treatment. In our first series of experiments, we'll look at ways to measure apoptosis via caspase activity in vitro in cell culture and some tissues using FLICA. FLICA stands for fluorescent labeled inhibitor of caspases and consists of three key components. There's a reporter tag, which is linked to a caspase targeting sequence like YBAD, and an FMK reactive group. So if you want to detect and quantify apoptosis in whole living cells, ICT's FLICA kits are a great option, and they're really easy to use. There's no lysis required. You don't have to permeabilize the membrane or treat the cells. You just add it directly to the cell culture media. It starts reacting in 15 minutes and can be incubated a few hours. You wash the cells, and then you can counterstain or take an image. The reagent lasts through many rounds of processing and dual staining studies, and they're compatible with GFP, DAPI, hooks, DTMR, antibodies, morpholinos, and you can analyze with a plate reader, a flow cytometer, or a microscope. So FLICA is an inhibitor, and that means that it's always fluorescent, and it binds with active caspase enzymes, which then inhibits the enzyme, meaning that it can no longer bind or cleave to another peptide. It becomes inactive. So FLICA is not cleaved by the caspase. It's always fluorescent, and once bound to the caspase, it is retained inside the cell, and the signal stays within the cells. Any unbound reagent is simply washed out of the cell. FLICA doesn't react with procaspases nor inactive enzymes, and as long as the cell membrane remains intact, you can specifically identify which cells are apoptotic or not. And these cells are ready for analysis and can be counterstained, fixed, or frozen. And it's really versatile. You can use it with all types of tissues and applications. And we have many options for FLICA kits to measure all the caspases or specific ones, and we're always developing more. So we have 3 reporters available in fluorescence, and that's carboxyfluorescein, and sulforhodamine in red, and 660. And which reagent you use to determine what's best for your experiment really depends on what other reagents you're using and what instrument you want to use for analysis. Here is a pretty easy, simple experiment using 2 reagents. And of course, there's a lot more data than what I'm showing here. These are cultured rat oligodendrocytes that were exposed to a drug and then labeled with green FAM FLICA and blue hooks. The top cell is apoptotic. Note the green staining for caspase activity and the fragmented nucleus with blue hook staining. There are also 2 live cells in blue below. In this experiment, they were testing a new drug to see if it would induce apoptosis in oligodendrocytes, which help insulate axons. And if the drug caused apoptosis, one side effect of treatment would be the loss of insulation around those axons, leading to a disease of the central nervous system. As only one cell was caspase positive, which is green, this particular drug was not a potent inducer of apoptosis. Its side effect may be less likely to damage the myelin sheath than other drugs. This is another easy, good model to study neuroprotection. In this experiment, also using FLICA and hooks, the researchers were looking for a way to induce oxidative stress and ROS, that's reactive oxygen species, production in neuroblastoma cells. They were able to do that by incubating the cells with hydrogen peroxide. Then they treated the cells with the drug to see if the drug would offer any neuroprotection from the hydrogen peroxide treatment by reducing apoptosis. Notice that the cell on the left is much brighter green than the cell on the right. It has more caspase activity. It also has a weaker blue signal from the hook, so we know the DNA has broken down further, so the green is easier to see on the left in the merged image below. Let's jump to a far more complex experiment in that they use 2 types of cells to understand how they interact and 3 different stains to measure 4 populations of cells. So I also put more data on the next slide. So in this experiment, the researchers at the University of Tokyo were studying mesenchymal stromal cells, that's MSC, derived from human umbilical cords. These MSCs may be used as a treatment and are anti-inflammatory and neurotrophic. So when injected IV intravenously, the UC-MSCs attenuate intraventricular hemorrhage-induced injuries. And then they see an increase in brain-derived neurotrophic factor, which is BDNF, and hepatocyte growth factor, which is HGF. And they wanted to know that will these factors help restore injured neurons? And yes, they did. So they harvested the neurons from embryonic day 16 mouse fetuses and cultured them without glucose or oxygen to cause an oxygen-glucose deprivation injury, that's OGD. Then they co-cultured the injured cells with the umbilical cord-derived mesenchymal stromal cells, which secrete the BDNF and the HGF. Then they labeled the cells with 3 stains, the green FLICA, the red propionimidide, and blue hooks. And they got 4 populations of cells. These are live, early apoptotic, late apoptotic, and necrotic. The living cells in the upper left, these are non-apoptotic cells that fluoresce blue with hooks, but otherwise remain unstained as they do not have active caspases, and they have intact cell membranes, which exclude PI. On the upper right, we have the early apoptotic cells. These cells fluoresce green with FLICA during the early stage of apoptosis as caspases are active, so they're blue and green, but not red as the membrane is still intact. On the lower left, we have cells in late apoptosis. These cells fluoresce green and red because they have active caspases and have lost their cell membrane integrity, so the PI stains them, and any blue from the hooks becomes more scattered. On the lower right, we see necrosis or death. These cells fluoresce red from PI due to the loss of membrane integrity. Then they counted the cells and quantified this. So let's take a look at their stats on the next slide. They calculated the ratio of apoptotic and necrotic cells to the total number of cells. There were significantly more apoptotic and necrotic neurons in the oxygen-glucose deprivation injured neurons, like in C, compared to the control neurons we see in B. They also found that adding neutralizing antibodies against the HGF in E and against BDNF in F reduces protective effects. So they know that HGF and BDNF offer neuroprotection. This is another easy in vitro experiment using ICT's red FLICA kit to study cryopreserved primary neuronal cells that came from the rat brain and were thawed and grown in cell culture. The goal here was to show the sensitivity of cryopreserved rodent brain neuronal cells for a plate-based cell culture assay of drug-induced neurotoxicity. Canic acid was used to induce oxidative stress, and the cells were stained with red FLICA to show the apoptotic neuronal cells, which responded as expected, making this a good model to study neurotoxicity. For something completely different, this experiment with zebrafish is really amazing for many reasons. They use double transgenic fish to look at neurodegeneration and the immune response to see if it's intrinsic or extrinsic. These were larval zebrafish that express red M cherry in the motor neurons. Zebrafish are really a favorite model to study regeneration and can make a complete recovery after spinal cord injury and regain all locomotor function. And these zebrafish were treated 72 to 78 hours post-fertilization with a control, DMSO, or a drug, MTZ. That's metronidazole, MTZ, I'll call it. And then they simply added FLICA, which you can use FLIVO as well, directly to the water. And that entered the whole animal and successfully labeled the apoptotic neurons green. So they labeled the whole live fish. Then they fixed the animals and mounted them in glycerol, all while preserving the green and red signals. This shows that MTZ leads to apoptosis in spinal cord motor neurons in these transgenic larvae. MTZ treatment leads to the ablation of all transgene-expressing cells and to microglia and macrophage activation, which leads to caspase activation and apoptosis in the green in the bottom right. These are whole mounts of a larval sea lamprey brain to see how it recovers after a spinal cord injury, SCI. So like zebrafish, lampreys have this amazing ability to completely recover from a spinal cord injury, which we did a webinar on earlier this year. So I'm reusing the slide. Lampreys have relatively few nerve cells and a few simple neural networks that were easy to study. Lampreys also have many visible neurons, and their neurons can survive for a long time in vitro and be easily labeled with ICT's reagents. So in this experiment, they wanted to detect neurons with caspase activity, and we're looking at retrograde degeneration with DTMR, which is red, to see if FLICA would influence the growth pattern, and found that it did not. This experiment was done by transecting the spinal cord and labeling with red DTMR at the time of injury. Then the animal recovered for 2 weeks, the brain was dissected, then it was labeled with green FLICA, you can also use FLIVO, and then fixed in 4% paraformaldehyde. Then they put it in 70% ETOH and froze it at -20. So they found that FLICA and DTMR techniques are compatible. In this experiment, also with lampreys studying spinal cord injury, they found that the molecule Rho-A could be a treatment for injuries. These researchers from Shriners Hospital investigated Rho-A, which is a small GTPase that inhibited retrograde apoptosis after spinal cord injury in H. Here they used the red FLICA product, SRVADFMK, that's sulfarotamine, with a green fluorescein tag morpholino, which is a type of oligomer used in molecular biology to modify gene expression, and an antisense oligonucleotide, those are the MOs. And the MO was applied at the time of transection, and then the animals recovered for 2 weeks. They dissected the brain, labeled with SR-FLICA, fixed, and imaged. And so as expected, if Rho-A is blocked, apoptosis decreases. The red fluorescence drops, which you see in H. The Rho-A knockdown is significantly reduced FLICA labeling. And so they know that Rho-A is involved in retrograde neuronal death after a spinal cord injury, and that Rho-A knockdown is beneficial for neuronal survival after a spinal cord injury. In this section, we'll look at live animal studies using FLIVO, which is an easy method to detect and quantify apoptosis in live animals in vivo. So FLIVO stands for fluorescence in vivo. You just reconstitute it, dilute, and inject it into the animal for 30 to 60 minutes prior to imaging or sacrifice. And like its sister product, FLICA, FLIVO is an inhibitor that's always fluorescent and binds to active caspases. So once it's bound to the caspase, it remains inside the cell. Any unbound reagent is pumped out of the circulation of the animal. So as long as the cell membrane is intact, you can specifically identify which cells and tissues are apoptotic or not. The animals and tissues are ready for analysis and can be counterstained with other reagents and fixed or frozen if you want to. We currently have 4 options for FLIVO and are always developing more. So they're using carboxyfluorescein, which is green fluorescence, sulfarotamine, and red. And for optical imaging, we have 2 near-infrared reporters using 690 and 747. FLIVO is really super versatile for use with all types of tissues, animals, and applications. So here we're looking at brain sections. FLIVO crosses the blood-brain barrier and was used to study brain damage in diabetic rats. On the top is the control rat, and on the bottom is the STZ diabetic rat that's 8 weeks old. 30 minutes before sacrificing the animals, FLIVO was injected IV, and then 20 micron frozen sections of the periaqueductal gray, the PAG, were prepared and counterstained with NISL, which is red fluorescent, to identify all neurons. Dying apoptotic neurons in the brain section appear yellowish-green with FLIVO, and in this model, they found that diabetic animals show greater levels of caspase activity in the PAG than control animals. So yes, diabetes causes brain damage, and the periaqueductal gray controls your pain sensation, and this area dies in diabetic rats. So in addition to peripheral neuropathy in the fingers and toes, diabetics may also have neurological problems processing those signals in the brain. Here we're looking at a flat mount of the retina, not the brain, and I wanted to include this because this experiment was done in almost the same way as the previous slide. It's a diabetes model. These are adult Sprague Dawley rats that were injected with STZ to induce diabetes and then sacrificed on day 30. Our green FLIVO was injected in the tail vein 30 minutes prior to sacrifice, and then the retina was harvested and fixed in 4% paraformaldehyde for 20 minutes and processed to create the retinal flat mount of the ganglion cell layer, that's the GCL. They also used the red stain NISL to label all neurons red. FLIVO is the bright greenish yellow spot below in B. So these researchers at Wayne State were studying diabetic retinopathy in the ganglion cell layer and the role of TXNIP, which is thioredoxin interacting protein. And this data supports that TXNIP is pro-inflammatory and pro-apoptotic, and it mediates neuronal apoptosis by activating caspase 3 and gliosis by upregulating GFAP, that's gliofibrillary acid protein, which is increased in the diabetic rat retina and is considered to be an early sign of retinal injury and the start of disease. Next, I'll show you another retinal flat mount using GFP. I'd like to show you one more image of the eye, and that was labeled in vivo with our red FLIVO probe because that is compatible with GFP, which is green. This is a mouse model of glaucoma looking at the microglia in a retinal flat mount. And microglia are immune cells, so they're not neurons. But I really thought this was interesting because the microglia cause damage to the optic nerves in the retina. And in this study, they wanted the microglia to become apoptotic and die so they wouldn't cause further damage. In these animals, the microglia express green GFP, and the researchers at the University of Utah wanted to know if radiation would help glaucoma by reducing neurodegeneration, and yes, it does. Since they're using GFP, they needed a red stain for apoptosis, and because they did not want any artifacts from euthanasia and histology processing, they used our in vivo apoptosis kit, SR-FLIVO, in red, which is sulfurotamine. Then they irradiated the mice first, then 8 hours later they injected FLIVO in the tail vein, and another 8 hours later they sacrificed the mice. Then they made retinal flat mounts, fixed for 30 minutes, and counterstained with hooks, which is blue. So these images are close-ups of the optic disc. The control mouse on top in A did not get irradiated. The irradiated mouse is on the bottom in B. The irradiated retina shows numerous and conspicuous apoptotic microglia in the optic disc. They're showing that irradiation reduced microglia activation, and that is associated with reduced optic nerve and retinal neurodegeneration in this D2 mouse model of glaucoma. Let's get back to the brain. Here we're looking at brain again, but it's a house sparrow and was processed in a multi-step procedure that took several days and shows that FLIVO will tolerate lengthy processing, as you can nicely see the green. So as long as the cell membrane remains intact, FLIVO will stay inside the cell. This researcher was studying naturally occurring neuron deaths in seasonally manipulated songbirds and needed to create a positive control to artificially induce apoptosis in the sparrow brain. So cerosporine was injected into the forebrain of a female house sparrow to induce apoptosis. 20 hours later, FLICA was injected IV into the jugular vein and circulated 30 minutes. The bird was sacrificed via transcardial perfusion with heparin, saline, and 4% paraformaldehyde. The brain was post-fixed for 48 hours, embedded in gelatin, and cryoprotected in 10% NBS and 20% sucrose for 48 hours more. 40-micron slices of the brain were made on a freezing microtome. The sections were mounted onto slides and cover slips with prolonged anti-fade mounting. Neurons with active caspases fluoresce green, and this image shows one apoptotic neuron at 100x. So that neuron really went through a lot of processing. This experiment was also done in birds, and you can see a larger section of the brain. In this experiment, the researcher wanted to know if caspases play a role in brain development, as birds and humans use hearing and sound to learn songs or language. And the cochlea is the only excitatory input into the nucleus magnocellularis, the NM, which is the second-order auditory neurons in the chick brainstem. And if the cochlea is removed, the birds can't hear, and this loss of stimulation induces a high level of apoptosis in the NM part of the brain. FLICA was injected 35 minutes prior to sacrifice, and in the control bird on top, you can see a low level of uniform background staining. But in the experimental chick on the bottom, nearly every auditory neuron in the deafferented NM is brightly stained with FLICA. So caspases are activated in every neuron in the NM just 6 hours after the cochlea is removed. And if the chick can't hear, the brain dies. Here are whole animal images using FLICA to study brain infections. This picture is taken with a Carestream in vivo FX Pro imager. These are adult wild-type BALB/C mice that were either given a control, 1 and 2 in the top and middle, or an intranasal inoculation with the HSV1 virus in number 3 in the bottom, which is absorbed to the epithelium and is known to induce apoptosis in the brain. 7 days later, at the peak of infection, the mice were injected IV in the tail vein without any reagent, number 1, or with NIR690, which is number 2 and 3, and then imaged 4 hours later. We can see strong caspase activity located in the brain of the bottom animal treated with HSV1 and injected with NIR690 in the bottom. The minimal signal was detected in the liver region of both mice injected with NIR690, that's number 2 and number 3, as the liver is the route of clearance for FLICA tracers. There is no signal in the unaffected control, which is number 1 on the top, and therefore number 2 in the middle reveals background staining. So this experiment uses 690, and they also did an experiment using 747, which I'll show you next. So these again are whole animal images from this experiment in color using NIR747, and includes the optional 747 free dye control. So in this experiment, again, these are wild-type BALB/C mice, were either given a control, 1 and 2 on the left, or an intranasal inoculation with the HSV1 virus, number 3 and 4 on the right. 7 days later, the peak of infection, mice were injected IV in the tail vein with FLICA NIR747, which is number 1 and number 4, or no reagent in number 2, or with the 747 free dye control in number 3. Then they were imaged 7 hours later with a Carestream in vivo FX Pro imager. We can see strong caspase activity located in the brain of the animal treated with HSV1 and injected with NIR747, that's number 4. There's minimal signal was detected in the liver region of the HSV animal injected with a free dye control, that's number 3, of the uninfected mouse injected with NIR747 in number 1. The liver is the route of clearance for FLICA tracers, and 3 of the mice with the red tails were injected with some reagent, that's 1, 3, and 4, where a reagent is likely to pool after IV injection. So this gives you an idea of what happens with all the controls included. In our next section, we'll look at experiments on mitochondrial dysfunction in vitro, which may be an early indicator of neurodegenerative pathology. ICTA has several assays to look at mitochondrial health, including MitoPT using JC1, TMRE, or TMRM. Here, the MitoPT JC1 reagent fluoresces either an orangey red when aggregated inside healthy mitochondria, which has a negative charge. And if the mitochondrial membrane has been permeabilized, the reagent will be dispersed in the cytosol and changes to green fluorescence. And it's very easy to use. Just add to the media and wash. This is flow cytometry data. In this experiment, they were investigating lidocaine, an anesthetic, which blocks neurons from signaling the brain and leads to necrosis and apoptosis. They wanted to know if the antioxidant N-acetylcysteine would block the damaging effects of lidocaine, and yes, it did. So here they're using the MitoPT kit, JC1, to measure mitochondrial membrane permeability. So in B in the middle, the data shifted to the right and is dropping, whereas the control in A looks similar to the NAC treatment in C. So therefore, NAC ameliorates the effect of lidocaine. In this experiment, they also used Human Neuroblastoma GILIN and SH-SY5Y cells and the antioxidant NAC. However, their research was looking at the effect of a drug, bortezomib. Not sure I got that right. They're looking at the drug over time and trying to see if NAC would reverse the effects. Again, that's an antioxidant. And yes, it does. So bortezomib is an effective inhibitor of neuroblastoma cell growth and angiogenesis. And here we're looking at a graph of the mean percentage of healthy cells. They have polarized mitochondria and fluoresce orangey red and are healthy. They found that, yes, bortezomib causes a time-dependent decrease in mitochondrial membrane potential, meaning that the red fluorescence decreases. However, NAC, that's the fourth bar with stripes, increase the percentage of cells with polarized mitochondria. The other MitoPT kits use TMRE and TNRM, which are also orangey red fluorescent probes. However, these probes fluoresce when they're inside healthy intact mitochondria, but then they don't fluoresce when dispersed in the cytosol. So you see the signal drop in dying cells. Here again, these researchers are using HT22 cells to study oxidative stress. Rather than looking at the effects of a drug, they were looking at gene expression. They were studying the gene CACNA1C, which shows a strong genetic risk factor for affective disorders such as depression and bipolar disorder, etc. They also used mouse HT22 cells and exposed them to glutamate to induce oxidative stress. And they found that silencing the gene reduced oxidative cell death, and downregulation promotes mitochondrial resilience to oxidative stress in neuronal cells. This is the final section of just a few slides. I'll show you how to assess apoptosis and lysosomal health using ICT's Magic Red Substrates. These reagents are substrates, so they are actually cleaved by the enzyme and they don't fluoresce until they are cleaved. So you can watch the color develop over time, or you can take a quick snapshot without having to wash the cells, although most researchers change the media. Again, these are very easy to use. Just add them to the media and image. In this experiment, they used the Magic Red DEVD substrate to quantify apoptosis via caspase 3-7 activity. They found that overexpression of GRG5 by embryonic stem cells leads to apoptosis resistance. Treatment with etoposide, which you see in the middle pictures, induces apoptosis in the control, in the upper right, while overexpression of GRG5 reduces apoptosis. That's less red on the bottom right. And that was quantified in the bar graph as 80% of the controls were apoptotic compared to 20% of overexpressing GRG5 cells. So overexpression leads to apoptosis resistance. This is another neat experiment using transgenic mutant neurons that are a model for Alzheimer's disease. These cells have defective lysosomes, and the researchers wanted to understand what was going on. They used ICT's Magic Red reagent for cathepsin B to monitor lysosome functionality. And they found that cathepsin B decreased in lysosomes in mutants, that's the lower right, compared to the wild type in the upper right, and that is quantified on the graph. So you get some pretty nice pictures of the GFP and the wild type compared to the mutant cell line. In this experiment, using MN9D neurons to study Parkinson's disease, the researchers were trying to figure out how MPP worked. They found that MPP is neurotoxic. It causes cytosolic calcium to rise and overload, which impairs autophagy and increases the lysosomal pH, which decreases cathepsin B activity. They found that treatment with calcium buffers like BAPTA-AM, which you see in C, the third picture, restores normal cathepsin B in A, which you see in the control, and ameliorates MPP activity. So yes, buffering of cytosolic calcium preserves the autophagy lysosome pathway caused during MPP-induced neuronal cell death. This last experiment is particularly cool because they grew the neurons in a microfluidic device so they could see the axons clearly, then tested for lysosomal health via cathepsin L activity. They also looked at cathepsin B, but I didn't show that here. So they found that if the degradative lysosomes aren't delivered to the distal axons, the neurons tend to die. And here they show that cathepsin L is working along distal axons in the control on the left to maintain local degradation capacity, whereas treatment with vancomycin A blocks cathepsin L activity, which you see on the right. So they were able to specifically live image these vesicular structures containing the cleaved cathepsin L in those distal axons. Pretty cool. Well, I hope this webinar was helpful for you. In summary, ICT has many fast, easy, and reliable methods that can be used to evaluate cell viability and functionality in neuronal studies in vitro, in vivo, and ex vivo. And they can be detected at many wavelengths like red, green, NIR, and 660. So which reagent is best for your experiment all depends on which instrument you're going to use for analysis and what other stains you want to use with it. All of these kits are available online, so please check them out on our website and expand your research toolbox by including ICT's reagents for cell viability. That wraps up our presentation for today. If you have any questions, feel free to send them through the chat box or to email help@immunochemistry.com, and we'll respond directly to you. Of course, you can always call us. We love hearing how you are using products and helping you set up your experiment. Again, thanks for tuning in today and for all the research you're doing. And thanks again to Allison Westcott, Dick Weninger, and Kristen Pauley, our marketing team, for managing these webinars. We here at ICT are proud to be part of the medical research community and provide these research reagents and kits that can help understand and someday cure neuronal diseases. I'm Sally Head-Dahlquist, President of Immunochemistry Technologies, and it's been a pleasure helping you find a cure.